All right, everyone. Happy Wednesday morning. My goal today is to try to get through some of the tissues that we need to talk about for week 2. And I wanted to draw your attention to week 3. In there, I added a practice quiz. So before you take the quiz for points, I wanted to give you a test of how I ask questions, what I normally am looking for, and it's kind of a test to make sure you can access the Blackboard quiz system. It is an optional quiz. There's no requirement that you take it, but if you want to have an idea before quiz number 1 in the midterm, how the content might be, what's the good word to put it, might be assessed, this would be a way to give you that opportunity. Okay, and I put that together yesterday, so it just showed up in the week 3 content link. All right, so tissues. Let's talk about tissues. So once we get to a place where cells are going to gather together and then certain cells are going to specialize to perform a specific function, we now move in the level of complexity up a notch and we start to see tissue types develop. And in the human body, we have 4 distinct tissues. We have the first one, epithelial tissue. Epithelial tissue is going to be any place where we are potentially going to interact with the outside world. So it's going to cover any exposed surfaces. So look at your skin. Everywhere on your body that is interacting with the outside world, that is a type of epithelial tissue that you see. It's going to line any internal passageways. So think about the entire gut, the alimentary canal, from mouth to anus. So everywhere that food, liquid, boluses of, somewhat digested material is moving through, technically it is outside the body. It's inside that tube, and that tube is lined by this protective epithelial tissue. The bladder. At some point, you start to collect urine. Urine is a very acidic fluid that contains all these waste products that we actually want to remove from the system. And the bladder is actually this balloon-type sac that collects enough urine until you get to a point where you want to expel it. So it is actually going to be an internal chamber that is lined with epithelial tissue. All your blood vessels are lined with epithelial tissue, as is the chambers of the heart. The nephron of the kidney is lined by epithelial tissue. The ureters that bring urine from the kidneys to the bladder. The oviducts that connect the ovaries to the uterus. The uterus, everybody's potentially heard of endometriosis. That's because the endometrium, the innermost lining of the uterus, which is an epithelial tissue, is going to be inflamed, going to be problematic. And then the epithelial tissue is also going to form glands. So when we think about sweat glands, ultimately sweat glands are going to be collecting water from cells lining the sweat tube, and that tubing is going to end on the surface of the skin. Think about the mammillary gland, so the breast tissue. The breast tissue, the cells line around these duct systems, these tubing systems that come together towards the nipple, and as the cells release milk solution product, that will then potentially collect in that tubing and then be squeezed out of the breast tissue when you are lactating and breastfeeding. So most of your glands then are formed of epithelial tissue because most of your glands that excrete secretions have some type of tubing that accompanies the glands to collect all of those secretions. All right? So that's our type of epithelial tissue. All right, connective tissue is actually going to be tissue that connects the other tissues to each other. So it's going to provide some of the support between the tissues. It's going to fill some of the internal spaces between other tissues. It's going to be our fluids of blood and lymphatic, so it's going to help transport materials from one place of the body to the other. It's going to provide strong frameworks because it's bone and cartilage, and adipocytes fall into this category, so fat tissue, so it's going to also store energy. In some ways, I think of connective tissue as like the catch-all category. It doesn't really fit in muscle, nervous, and epithelial, so it kind of gets thrown into connective tissue. So connective tissue is a lot of different things, but the main point is they are usually between the other tissues and are somewhat the glue, the structure that helps connect skin to muscle and neurons into the system so they can interact with the other tissues, okay? All right, our third type of tissue is muscle tissue. So all of the cells that are part of muscle have this ability to contract. So the cells specialize in a way that they make large amounts of proteins that upon acting together in the presence of calcium and energy form excitation, contraction, interactions, and can change the shape of the cytoskeleton and thus change the shape of the cell. And we can then, based upon our structural manipulations of how these cells are oriented and how we connect them to connective tissue, such as bone, we can potentially use the force created by the shortening to generate movement and to make that force work for us and create different changes to the body. All right, so the key is somehow, some way, the cells develop this ability to change their size, their shape, and they orient themselves in a way that doing so in a large number and a large group lets us, in physical terms, focus that energy, that force, in a way that we can then use it to make movement or move things through our pipes and tubes and keep pumps going, so like the heart, okay? Now, nervous tissue is going to be two types of functional cells. All right, there's all the supporting cells, that's the neuroglial cells. So the neuroglial cells typically are the high-numbered supporting cells that keep the neuron, the main component of nervous tissue, alive and operating. And nervous tissue cells, the neurons specifically, this is the cell that has the unique ability to carry electrical impulses. So it has this ability to manipulate the outside and inside ion concentrations, and in doing so, generate an electrical change across the membrane. So those action potentials and graded potentials, and it has an ability to in some ways integrate and determine and understand whether it should pass on the electrical signal or not, and then when it passes on the electrical signal, the electrical impulse, the action potential, it has this ability to then convert it to a chemical signal so it can continue to propagate to other tissues and other cell types. Okay? So those are our four tissue types. So epithelial is going to be any place where we are potentially going to be involved with touching, coming into contact with the outside world, or coming into contact with fluids or things moving through a tube. And as they move through a tube, per physics, there could be turbulent flow, there could be laminar flow, there could be stress applied to the walls of that vessel or that chamber. Okay, connective tissue is a catch-all category. Lots of different types of things fall into this category. They provide structure, they provide framework, they provide glue and storage capacity. Muscles, everything in these cells, special proteins, special organelles are there that allow the muscle to contract. Nervous system, These neurons within this tissue have this specialized organelles and channels and ions and properties that allow it to have an ability to manipulate ion concentrations inside versus outside, make changes in small microseconds, and that ability then allows them to carry electrical impulses down their membrane. All right, so let's focus on our barrier system. So when you think about epithelial tissue, it's barriers or it's glands. Barriers or glands. All right, so when we talk about epithelial tissue as typically barrier cells, cells that are going to be the first line of defense to protect something from the outside world from getting in, some stress outside the body from damaging inside, we call it epithelium, epithelia. Okay? And there's 5 important characteristics associated with tissues that's a barrier. So the first thing we usually see with barriers are cells are touching each other. There is little to no space in between the cells. And in many cases, anchor proteins are going to bind the cells to one another to make the cells and the space between them more impermeable. So cells are touching, cells are trying to make a wall that differentiates outside versus inside. Now, because of the cells having this one side of themselves exposed to a fluid, to the outside world, to acid urine in a bladder, there is a difference between the membrane interacting with the outside world versus the membrane of the cell interacting with the inside and connecting to the body. And so because those membranes are not exactly the same and they don't have potentially the same membrane proteins and receptors and antigens and everything else, they have a polarity, they have a difference. So one portion or side of the cell exposed is going to be very different than the side of the cell that's attached to the body and anchoring that cell so it doesn't collapse, fall off, or get pulled away. Okay? Now, the cells are touching. The cells are anchored to each other. The cells are trying to be a barrier. So one of the things that you will not see in epithelial tissue is blood vessels. You won't see capillaries between the cells. So these cells are avascular. And because they're avascular, they have to get all of their nutrients from the part of the cell that interacts with the attached underlying tissue. And so that attachment to the underlying tissue is usually going to be called some type of basement membrane. You might also be calling it a lamina propria, some type of connecting glue that keeps that epithelial cell that's exposed to the outside world connected to the underside. And it's in that basement membrane, that lamina propria, that we tend to see blood vessels, so the nutrients can then diffuse from the blood into the connective tissue of the basement membrane and then diffuse into the cells sitting on the basement membrane. All right? We're going to see any type of nervous receptors, nerve endings that could potentially do itch, pain, discomfort, stretch. They're all going to sit in that basement membrane. So that basement membrane, in addition to being the glue that connects and holds the epithelial tissue to the body, is also the site where all the other tissues can sometimes worm their way up and then get really close to the outside world and potentially sense and interact with the outside world too, but with a little fine barrier, or in some cases a very thick barrier between them. All right, these epithelial cells are subject to potentially radiation, convection, changes in temperature, abrasion, tearing, so lots of different things. And they have to constantly replenish the cells that are damaged or sloughed off or pulled off or lost to the outside environment. And so one of the things we'll see in epithelium is that they undergo a very high mitosis rate. So they are constantly, some of these cells are constantly in a state of mitosis. So there's some in interphase, but a large portion of the cells are always going through mitosis. And going through mitosis and generating 2 new daughter cells allows constantly the tissue, the barrier, to be what do you call it, remade, and any damaged or lost cells to be recovered, okay, or not recovered but replaced, all right? So that is one thing you should see when you're looking at epithelial tissue, these 5 characteristics. All right? Now, glandular epithelial tissue, you may see a lot of these same characteristics, but in many cases, the cells are going to be around some type of tubing, and the tubing is collecting secretions from these cells that then are, in like the pancreas situation, secretions or digestive enzymes are released into the small intestines. In the stomach, the pits, the glands made here, release secretions into the pit itself, and then it's released into the stomach proper to interact with the food. Salivary glands, there's tubes that connect it and release the saliva into the mouth and underneath the tongue. Right? Sweat, they release it on the skin. Earwax, they release it in the ears. All right? And so those secretions, even though there's a tubing system, the cells around the tubes kind of act like epithelium, but they make secretions, which epithelium cells don't always make. Don't always make. They can make. All right? When we look at epithelial tissue, we tend to look at 2 big factors. We look at, is there a multiple layer of cells on top of the basement membrane? So do we see stacking of cells, and the further away from the basement membrane, the more likely that the cells are dead? So the ones potentially touching the outside environment might be dead cells. Look at your skin. At any point in time, you know, if you're rubbing the skin, you exfoliate, you're tearing off dead cells that have moved so far away from the basement membrane, and therefore the capillaries and the nutrients, that they've died. Okay? So we look at whether things are multi-layered or single-layered. Right? There are many places in our body that, while we want a barrier, we want that barrier to be as thin as possible. So we want single layers because we don't want to have a increased diffusion distance or increased amount of space that things would have to move to be able to get from outside to inside the body. So the lungs are a good example of that. When you look at your alveoli, your air sacs, you want a very small space between the air and the blood. And the blood's flowing in capillaries. So your epithelial tissues lining the air sac, lining the capillaries of the lung capillaries, are actually very thin, single-layered cells. Okay? So that's some places that we look at. So in the mouth, it makes sense that when food comes in, and food has different textures, and as we mash and tear and move around with our tongue, we don't, if we were to bite the tongue or bite the lining of our mouth and our cheek, we don't want that to be single-layered. We want it to be multi-layered so we can handle the stress of chewing. The nose, same thing. As we bring air and potentially have to blow our nose or pick our nose or whatever, we don't want single layered. So those are areas we want lots of barrier, lots of epithelium to be multiple layered, so we increase the likelihood that that if a little damage occurs, there's multiple layers that can compensate. Right? So anytime you have a multiple layered, you're typically going to see stratified in front of the cell, the cell word that kind of describes what the cell looks like. Okay, so stratified always implies multi-layered. Simple implies single-layered. All right. The next way we classify epithelial tissue is you look at what shape, what is the morphology of the cell. So there are going to be cells that look like flat crepes, like really fat pancakes, fried eggs. They have a little bit of a raise maybe where the nucleus is, but for the most part they are thin and they are flat. That shape is that looks like, again, a fried egg or a flat pancake is a squashed cell. And because it's squashed, we're going to call it a squamous cell. Right? And then we're going to see cells that are nice and square shaped. They look like cubes of sugar. And so because they look like cubes, we're going to call them cuboidal. And then we're going to have cells that are going to be long and skinny. And so the part of the cell interacting with the outside environment is going to be kind of short. It's going to be one of the widths. And the part anchoring it to the under part of the body is going to be short. But it's going to have these long parts, the rectangular long parts, are going to be connecting to the other cells. And so those are going to be our columnar. All right? Now looking at this slide, we see two unique little names here. We see pseudo-stratified columnar and transitional. All right, pseudo-stratified is a way to say it looks and it fakes you out to think it's stratified. So it's really single layer. But because the nuclei don't line up, because there's constantly regeneration, and it gives sometimes that appearance of mitosis and multi-layers, it looks stratified, but it really isn't. And you find this type of pseudo-stratified columnar ciliated epithelium in the trachea. And let's just think about the respiratory system. The mouth and the nose are the beginning of the respiratory system, and then you work your way to the back of the throat, into the trachea, into the bronchi, and eventually all the way down to alveoli. So if you think about the passageways, you go from stratified at the opening, and you need to go to simple squamous at the very end of the tubing, at the very end of the alveoli. And in that transition, you got to go from multi-layered to single-layered, and you have to basically make a change in the lining of the tube. And so pseudo-stratified columnar is part of where that change goes from the pharynx, the larynx, the upper regions, the mouth, the nose are stratified, and the inner lower regions are simple. And so pseudo-stratified is part of that process. All right, transitional, this is in the bladder, okay? The bladder. Now the bladder is going to be an organ that when it's full of urine, it actually stretches. And so the cells, when it's full of urine, that are touching the acidic fluids that are full of ammonia and excess proteins and excess ions, and excess potential acids like hydrogen ions, it's going to need to change its shape depending upon if there's fluid inside the sac or no fluid inside the sac. But in addition to that, it needs to be multi-layered, because think about it. Acid could eat away and break bonds of proteins embedded in the membrane of a cell. So, I mean, that's how we break proteins down in the stomach. We use acid. So we need multiple layers to line the bladder so that the layers actually touching the urine, as they get damaged, you know, they can be constantly replaced, and we don't potentially have urine leaking into the lower pelvic cavity. But we need these cells to take different shapes depending upon if the bladder is distended and full or empty and collapsed. And so that's why they call it transitional. So when it's distended and the bladder's full, the cells may look like stretched out squamous cells. And then when it's not full, they look like big, fat, you know, circular blobs. Okay? So let's look at some pictures. All right? So again, anything simple is single layer. Cells are, let's go through those characteristics. Cells are touching. There's very little space between all of our cells. The cells are going to have a side exposed to the outside world, and so the membrane out here might be very different from the membrane anchored to the basement membrane or lamina. So we would expect maybe lots of anchor proteins down here versus up here. We might expect to find lots of proteins to interact and give feedback of what is the fluid in the tube doing, or what is the air in the outside world doing to the body. Right? There are no blood vessels in any of these tissues, okay? And because you're constantly sloughing off, losing, getting damage incurred to the layer touching the outside world, you have to constantly be generating new cells to replace them. And that's what usually the innermost layer of any stratified tissue is going to be mainly composed of, mitosis-rich regeneration occurring. Okay, so draw your attention again, the cells, simple flat pancakes, simple squares, simple columns, right? Multiple layered, mostly the cells are flat pancakes. They are maybe a little squarish, columnish down here, but most of the cells in the entire tissue are squamous, so stratified squamous. Most of the cells here, square, stratified cuboidal. right? Pseudostratified. So look at this versus simple columnar. The nuclei do not line up. The cells are squishing and squeezing and pressing on each other, and so the nuclei can end up high-low, and because of that, it gives the impression that there's a layer here and there's a layer here, when in actuality, it's all one layer, right? So that's why it's pseudostratified columnar. Transitional, again, in this case, I would say the bladder is not distended, so the cells are more puffy. But when the bladder gets pressed and distended, the cells are going to be more flat and squamous looking. Right? So where do we find simple squamous epithelium? You're going to find it in places where you have little cavities to reduce friction. So the heart and the lungs in our thoracic area are movable organs. And we don't want the heart to rub up against the breastbone and the ribs and the muscles that are part of the thoracic cage. We also don't want the lungs to rub up against them. And we don't want them to rub up against the spine and the esophagus and the trachea. So in some ways, we wrap the heart and we wrap lungs in a little bit of a fluid-filled sac. And in making that fluid-filled sac, if you look at it, there's simple squamous epithelium. Then there's a basement membrane known as areolar tissue, a type of connective tissue. And then you actually see muscle. Okay? And on the other side, the chamber of the heart, you see simple squamous epithelium, which is now known as endothelium, and you see a basement membrane, areolar tissue, and then you see the muscle. Right? So when you think about it, the heart, you never actually have the heart cells interacting directly with the blood, and you never have the actual heart contractile muscle cells touching the bone and the muscles of the thoracic cage, and they never actually touch the lungs. And so the simple squamous epithelium that's going to form the membranes that make the sac, which will then be the pericardial cavity or the pericardial sac. You're going to have the mesothelium and the areolar tissue, and then the simple squamous epithelium lining the heart chamber, and then eventually lining all blood vessels is known as endothelium. Okay. Other places you're going to see simple squamous epithelium is going to be lining the capillaries of all of your capillaries in your body and lining the alveoli of the lungs. Okay? And if you look at the microscope, if you look at kind of like the heads-up display, so if this was the alveoli or something, you would basically see flat pancake cells. Now, you can't 100% know that you're looking at simple squamous because you don't know, do they go long underneath or square underneath. But if you look at them in cross-section, you'll see they're very thin, very thin, very thin. Okay? All right. Stratified squamous epithelium, the best place you see this is the mouth, the lungs, the vagina, the skin, any place where you want, look at it, multiple layers. Majority of those cells, flat, dead, squashed cells. Innermost layer is where you might see more squarish cube columnar, but there's lots of regeneration. Lots of regeneration means lots of nuclei, so that's why it's a little bit darker purple. All right? No blood vessels. All right, so cells are touching, avascular, so all the blood vessels are in this region, in the connective tissue and the basement membrane underneath. And so as the cells move further away from the basement membrane, they die and they are more likely to then be sloughed off. Okay, now there are some places in the body we want the stratified squamous cells to be waterproof, and then in other places not. So the vagina, the mouth, the nose, we want it more waterish, water-friendly, because we want to keep things lubricated, less friction, less abrasion, less cuts, less tearing in those regions of the body. But the skin, we want it to be more waterproof. So tend to be areas where we want waterproof, there's more protein content called keratin present. And you'll know that keratin is a big part of your nails and hair your hair are very, very waterproof. Very little keratin in the simple stratified squamous epithelial tissue in your mouth, because again, you want mucus, you want to reduce cuts and abrasions in the mouth, and again in the vagina for coitus. Right? Cuboidal epithelium tends to be glandular epithelium. All right? So both simple and stratified cuboidal epithelium tends to be in glands. So you're going to see them lining all this tubing. You're going to see them releasing secretions into the tubing. The tubing will collect maybe in bigger pipes, bigger tubes, bigger sacs, and then released at the exit point, whether it's on the skin, at the nipple because it's a mammillary gland or a sweat gland or ear gland or a salivary gland. So most of your glands, if you were to dissect them, are going to be cuboidal type of epithelial tissue. Okay? So most of your cancers that come from the glands of your body, so the thyroid gland, the ovaries, the salivary glands, the pancreas. So most of those cancers are actually derived from the cuboidal epithelial cells becoming inappropriate and becoming cancerous. Right? Transitional epithelium is going to be the cells touching urine. They're going to have a different shape depending if the system is stretched or not stretched, and so they are going to be multi-layered. Right? Columnar epithelium, those nucleuses are going to tend to line up. They are going to be found in places where you interact with food and you are involved in trying to get nutrients into our system. So and they're going to be in the uterine tubes. The little hairs here are going to help move the egg towards the sperm and move sperm towards the egg. Okay? So you tend to see cilia with columnar epithelium. You will also potentially find secretion cells known as goblet cells. So there might be a specialized cell that develops that then its whole purpose is to make mucus and make secretions that help the cilia move things up or back and forth. Okay? Pseudostratified is in the trachea. So the trachea is a big place, a little bit of that transition from where mouth and nose make their way to eventually alveoli, simple squamous. And it looks like a hot mess, but it's ciliated cells and typically going to be in those airways. Okay? Stratified columnar epithelium, you don't see this a lot, but in some of, again, that transition of where stratified squamous is slowly making its way to pseudociliated columnar epithelium. So in some of the transitions around the pharynx, the back of the throat, and the anus, and then again some of the ducts to help get rid of the secretions at the very end of the tubes for salivary glands, mammillary glands, and things like that. Okay? All right, again, your glands are going to either be endocrine or exocrine in nature. Endocrine glands make hormones, exocrine glands make secretions. Most of your glands are always going to be some type of cuboidal, right, epithelial tissue. All right, I got to go to class, so I'm going to stop here, and I will try to pick up with connective tissue, get through muscle and nervous tissue in our next recording. So look for that to either be later today or tomorrow after hopefully I get back from my orthopedic visit about my knee. All right, questions, please use the open forum, please use email, and keep working on those worksheets and getting your bearings, okay?